Okay, so now what we're going to talk about is we're going to talk about this thing called the sarcomere right up here. This is considered the functional unit of muscle tissue. That means that is the thing that carries out the function of that tissue. And so let me show you in a couple of places what this is so you can hopefully visualize it well. Okay, here is our muscle cell, our myofiber model, and so I just want to remind you that this whole thing here, okay, all of that is the muscle cell. This is the plasma membrane of the muscle cell. We see that plasma membrane also down here in pink. Each individual circle is a myofibril, and then each individual line, we can see the little circles that make up each of these myofibrils. You can also see a line going down from top to bottom. That is the myofilament. Okay. Now there's sections of this myofilament that work together. Okay. And so what you can hopefully see across here, you can see a line going this way across. Okay. And so from one of these lines that look like this down to this other one right here, okay, that is a sarcomere. So everything from here up to here, okay, is sarcomere. Now, if you notice, here's one sarcomere, here's another sarcomere, okay, so they're repeating units that are going to go down this structure, okay, and in the myofibril next door, you're also going to have a sarcomere, and then another sarcomere going down. Okay, so each myofibril is made up of these regular units called sarcomeres. And what we're going to do is we're going to get up very close to the molecular level. We're going to look at the molecules, the proteins that are there and what they're doing and how that helps the muscle cell contract. Okay, here's another view of a similar thing. So excuse me. What we're looking at here is going to be a myofibril. And we're now getting up close and we're looking at one section. So if you notice, here was that line I pointed out to you earlier. Here's another one. So from here all the way to here is a sarcomere. And so now we're looking up close. So here's that line, here's that line. Again, between those two lines is what's called a sarcomere. Now, as we're looking at the molecules here, the first thing we're going to look at is we're going to look at the proteins. And if you need reminding of what a protein is, go back to chapter 2. It's one of your macromolecules. Okay. So, what we have here first is we have this thing that looks like a zigzagged line here. This is made up of a protein called actinin. Okay, so over here another zigzag line. It's a protein called actinin. These red lines right here is a protein called actin. And you notice it's attached to actinin. It's also called, actin is also called the thin filament. Now here in blue is a molecule called myosin. It's a specific protein called myosin. And you've got them hooked up back to back. Okay, so here's one long myosin hooked up to another long myosin. Notice it has these little barbed looking things hanging off of them. Those are called myosin heads. Myosin is also called the thick filament because it's fatter. And then over here in green is going to be this thing that looks a little bit like a spring, and that's called titin. So what I'm going to do now is there's an active learning exercise where you can actually build your own sarcomere and practice then using it later on. So I'm going to switch to that now and show you how you can build your own sarcomere. Okay, so what I have here are the cutouts that are in the active learning exercise that is posted on website. Let me just go through, since the resolution of my camera may not let you see them. Here we have actinin in this zigzag line. The straight line is actin. Over here we have this thing with the barbed heads on it. That is myosin. And then this that looks like a spring is called titin. The wavy line is troponin, tropomyosin, and we're just going to move that off to the side for now. We'll use that later, but not right now. So my suggestion for how to begin building this is to first take your actinin. Actinin is the two boundaries of the sarcomere. And so you put that on either side. Now actin, the straight lines, are attached to the actinin. So that's what I'm doing right now, is I'm attaching the actin to the actinin. And in fact, I'm even going to tape this down because they are physically connected to each other. So we can tape that down because that interaction is strong, and as long as everything's normal, that connection is not going to be broken. Okay, now let's go ahead and put myosin in place. Now, myosin is set up so that it is back to back. And we are going to put a couple of myosins together. Now, again, the connection between the myosins is very strong. And so I'm going to tape those together. And in fact, there's this line that goes down the middle, and so I'm going to tape these together like that so that myosin is held tightly together. Okay? So, we're almost done with our sarcomere. The other thing that I want to put in place is this molecule called titin. Titin sits between actinin and the end of myosin. Okay, now if we look at this, we can ask ourselves how, if this is the thing that helps muscles to get shorter and longer, how might that happen? And what happens is that these things move past each other. So here we have a muscle that just got shorter, and here we have a muscle that just got longer. Okay, so again, this right here, the myosin, is a thick filament. Here in dark green is actin, that's a thin filament. And the way that this unit gets shorter and longer is the thick filaments move past the thin filaments. Now, what's the role of titin? What titin does is it really does act like a spring. And if this starts to get too far away, the titin acts as a spring that says, hey, actinin, don't keep going that way. Stay up here, at least this far away from myosin. Okay? And the same over here. If this actinin starts to get too far away, titin is going to pull it back up. Okay? So we never get too far away. Okay? And so this is how the sarcomere works. works. Okay, so now that we've talked about all of the different proteins that are found that make up this sarcomere, and remember a sarcomere runs from one zigzag line to the other. Now we have a bunch of different terms, and these terms are for regions of this structure. So we're no longer talking about protein names, we're talking about regions, areas within this structure. So again, here's the sarcomere that we've talked about again. So this thing that looks like a zigzag, we can call that a Z line. So the protein that makes it up is actinin, but the region is called the Z line. And here you see another one. This line where the myosins are connected back to back is called the M line. And you can think M for myosin. Now the A band is the region that extends from one end of myosin all the way to the other end of myosin. The H band is the region that contains only myosin. So if we look in this region, we only have myosin there. Myosin extends from here to here, but notice right here you have some actin overlapping it. So as soon as actin starts to overlap, you are no longer in the H band. The I band is going to be the region that only has actin or actinin or titin, but no myosin. So notice it starts right here. It would actually go across this Z line over to this region where the next sarcomere would be and where the next myosin would start. The sarcomere, as we said, extends from Z line all the way to the other Z line. And I'm actually going to give you one more region right here where you find both actin and myosin, so we can see it in this entire area here. That's called the zone of overlap. So if I was going to write that out, the zone of overlap. Okay, so what I'd like for you to do, is to now go to your notes and see if you can take this information that we've just talked about and put it in. Okay. So for example, sarcomere. How would you define a sarcomere? What is a sarcomere? And hopefully you should have something in that definition about running from Z line to Z line. We already talked about how the functional unit of muscle tissue is the sarcomere. Thin filaments consist of mostly actin. Thick filaments consist mostly of myosin. And then here's a picture. It's very similar to what you've seen before, but I want you to see if you can first label the proteins: actin, myosin, actinin, titin. Okay? And then see if you can label the regions: H band, A band, I band, Z line, M line, sarcomere, zone of overlap. Also make sure you can describe these things in your own words. Okay, so for example, an H band is going to be the region in which myosin is present, but not actin. Okay, so see if you can explain these in your own words. Now, what I'd like for you to do here is to describe what's going on when a muscle is contracting, when this sarcomere is contracting. Okay, and so I'm going to go back to the paper model that we created of the sarcomere and go through this with you. Okay, so here we're back at our paper model of the sarcomere. For the moment, I'm going to take Titan out of the picture. Just know it's there, what it's doing, but I think it kind of muddies the waters. Okay, so let's make sure looking at this that we can recognize our different zones and regions. Okay, so between my fingers here is the H band. That's where we have myosin, but no actin. From here to here is the A band, because it is the length of myosin. Okay, from here all the way over to the other side where we'd see a mirror image, okay, would be the I band, so we have actin present, but no myosin. All right, and the other two things we have, one is we have the entire sarcomere running from Z line to Z line. This is one Z line, this is the other Z line. Right in the middle is our M line, where the myosins are back to back. And then we have a zone of overlap right here, where we see both actin and myosin. We have another zone of overlap right here. We also see actin and myosin together. Okay, so as contraction happens, notice that here's our thin filament, here's our thick filament. And notice how they simply slide past each other. This is called the sliding filament theory, that for the muscle to get shorter or longer, the filaments slide past each other. Here we go shorter, now these filaments slide past each other again to get longer. That's all the sliding filament theory is. It sounds like it should be really complicated, but in reality, it's not. It's fairly simple, particularly once you understand the structure of the sarcomere. Now, let's ask ourselves, when a muscle gets shorter, what happens to all of these regions? Okay, so let's say here our muscle is at rest, and as we said before, in between my two fingers, right in here, is the H band. And we can even mark the H band at rest went from here all the way to there. Now, let's shorten our sarcomere. Let's make our muscle contract. What happened to our H band? Well, it's still the region where myosin is present and actin is not. So now it's between my two fingers here. Notice it's just this region. So if we were going to measure it now, we would say that it is from here over to here. So a relaxed muscle has an H band this long. A contracted muscle has an H band this long. So during contraction, the H band becomes shorter. Now, so what I would like for you to do, if you look here on your paper, is it says, when a muscle contracts, what happens to each of the following zones? The H band, that one gets shorter. So I'd like for you to go through each of these and see if you can use your model and figure out what happens to each one. I would also suggest that you add another one, which is the zone of overlap, and list what happens to that as well. Does it get shorter, longer, or stay the same length? Now there's a couple of questions here where it says if the thick filament hits the Z line, can the sarcomere continue to shorten? So let's use our model and figure that out. So we're back at our model. Okay. So as a reminder, here's our thick filament. Here's our Z line. So if we shorten to the point where the thick filament has now hit the Z-line. Can we shorten any more? And the answer is no. This thick filament physically cannot get past this Z-line anymore. It's hit a dead end. And so this is why our muscles can only shorten so much, because if those thick filaments hit the Z-line, there physically can be no more shortening. They can't get past. Now, the next question asks, if the zone of overlap is reduced to 0, can the sarcomere shorten? Okay, so let's remind ourselves of what the zone of overlap is. The zone of overlap is where actin and myosin overlap. Okay? Excuse me. So, what you need to know to answer this question is you need to know that in order to shorten, these actin heads actually will grab onto myosin and pull it back. That's called a cross bridge. We actually form a bridge between myosin and actin, and these heads pull the filament towards it. And that's going to happen on both sides, so you get this type of thing. So what that means is to get the process started, you have to have at least some overlap between actin and myosin, because that head has to grab onto actin. If all of a sudden we reduce our zone of overlap to 0, now there's no overlap. This actin head, it can't grab, this myosin head cannot grab actin. It physically can't reach it. And so if this sarcomere gets too stretched, then no contraction can occur because the myosin head can't reach actin. You must have at least a little bit of overlap so this head can reach actin and start to pull it back. So what I'd like to point out here that you have on your homework lesson is you do have a couple of videos that are embedded that give you cartoon animations of sarcomere structure and function, and here it's showing how the filaments slide past each other. So my suggestion is to also look through these videos and then go through and work through these activities to make sure that you have this information properly in your head. And again, please feel free to contact your instructor with any and all questions.